It's such a pleasure to be back for this year's annual forum, to be able to share ongoing progress being made in Waldenstrom's. And for me, this is now, I think, year 28 that I've been attending the forums, starting way back when Arnie Smokler first invited me to speak. And we have indeed made, incredible progress. And so in sort of this new modern era, I just want to talk to you just to tell you a little bit about where we were not too long ago. And this was the so-called pre-mid-88 era, before we had the discovery of the MYD88 mutation. I refer to this era as the hand-me-down medicines era, because all we did in that time was we waited for our colleagues in the lymphoma world, the CLL world, the myeloma world to be able to vet out all these new drugs and then eventually hand them down to us. And this was really quite a chore, because many times the side effect profile that had been reported with these drugs was very, very different than what we encountered in Waldenstrom's. And also the activity. We saw activity for some of these drugs, such as lenalidomide, pomalidomide, thalidomide in myeloma, but in Waldenstrom's they produced toxicity with very little benefit. So importantly, in 2003, at the second International Patient and Physician Summit on Waldenstrom's, which was held in Los Angeles, we began to strategize how we could get at the heart of discovering what caused Waldenstrom's, what was the genetic event that actually led to these cells becoming problematic. And you can see here we had quite a brain trust, including, you can see Zach Hunter way in the back, along with Andy Brannigan, Nikhil Munshi, a very young Nikhil Munshi there shown. But it was a wonderful think tank for us to strategize about what sort of technology we needed to employ in order to be able to discover the genetic events behind Waldenstrom's. And that came in 2011, employing whole genome sequencing. That was a brand new art at the time. Today, you can get it done anywhere. In fact, you probably see ads if you end up on the internet now telling you for a couple of $100 they'll do a whole genome sequencing on you. But back then it was very expensive, and more so, there were very few people who understood how to interpret this data, because it involved over 3 billion DNA molecules being analyzed, and you had to literally find the needle in the haystack. And this was funded by the IWMF, as well as by an incredible philanthropist and friend to our community, and that's Dr. Peter Bing, who at the time was the chairman of Stanford University. And so with his philanthropy, we embarked on this project. And at the time, once again, it was a very, very expensive proposition. But what we ended up finding was that the MYD88 mutation, in fact, explained as the key genetic event Waldenstrom's in about 95 to 97% of patients. And this was in the year 2011, reported as the best discovery at the American Society of Hematology, which is our key meeting in the hematology world. And it was reported then in the New England Journal of Medicine in 2012. So it was, in fact, a pivotal moment in the history of Waldenstrom's. Now, for the next 15 years, Dr. Guang Yang, who was in our group, began to try to decipher exactly what this mutation did. Unfortunately, I don't have a pointer that I can go through all these little signaling diagrams for you, but what I can tell you is it's a complicated process. MYD88 itself serves as a hub that signals in response to viruses, bacteria coming in contact with B cells. And when it actually is able to activate itself because of these encounters, it triggers this very complex pathway. Now, the thing is, if you have the MYD88 mutation, you no longer need that external signal. You don't need any more pathogen to come in contact with the cell. It just begins to fire. And as you can see sort of the pattern here, that one of the key events is that it turns on this molecule HCK. In fact, HCK shouldn't be there at all. But because of the MYD88 mutation, and we worked out all the kinks of this, largely with funding from the IWMF and LLS, but then more recently with the NIH, we found that HCK is upregulated, it's turned on, and in turn what it does is it turns on BTK. You can see BTK just south of HCK. And this is what actually opened up, our eyes to the development of all the BTK inhibitors. And of course, we'll talk a little bit more about this in just a second. So with the discovery of this connection of MYD88 to BTK, this allowed us to get the first ever breakthrough designation for fast track approval in any oncology indication. I want to say this is really a very special thing that all of us should be proud of, because this shows how science can be translated into advances in therapy. And then shortly thereafter, with the clinical trial results from the ibrutinib trial, we got the first ever drug approval for Waldenstrom's. And that was a very proud day. We were at the FDA in June of 2014. I was there along with my colleague Leah Palumba. Ranjan Advani at Stanford also participated in this pivotal trial. But you can see, in fact, we're all happy because we had very good news from the FDA that they wanted to help us get that first ever drug approval for Waldenstrom's. And then what ensued was all these clinical trials, because once you get an approval, every other pharmaceutical company becomes interested. And that's a good thing, Where were you? And this actually is a complex slide because it shows you all the different drugs that have been vetted in Waldenstrom's, including ibrutinib, zanubrutinib, acalabrutinib. In Japan, they've been doing trials with tirabrutinib, and it's approved there. And then more recently, pirtobrutinib. But I just want to summarize all these. It's nice to take very complex information and summarize it. But I'll tell you, as a single agent, these drugs are the most active in Waldenstrom's. So over 90% of Waldenstrom patients will get some benefit, major responses, which means at least 50% reduction in disease burden. You see in about 80% of patients. You also see what we call VGPRs, very good partial responses, which means at least 90% reduction in disease burden is measured by IgM, and that occurs in 30% of patients. So what you don't see here is complete remissions, and I want to come back to that point in just a second. Now, more importantly, look at that number underneath, PFS. That stands for progression-free survival. What that means is it's the time that the disease is kept in check without it progressing, and that the patients maintain benefit. And at 4 years, 3/4 of the patients in all these trials show this kind of aggregate benefit. So this is why we're very excited about the use of BTK inhibitors in Waldenstrom's. Now, there's also a second mutation, which I know many of you have heard of, and that involves CXCR4. This was also discovered in my laboratory, but this because of the prowess of Zach Hunter. Now, Zach looked at that same whole genome sequencing data, which others were finding very complicated to interpret, and he made a discovery which was very important. He rewrote the algorithm for how this data was being processed, and he found, in fact, that patients with Waldenstrom very frequently had these CXCR4 mutations, which I'll tell you about in just a second. This was a fairly unique event for Waldenstrom's because MYD88 mutations you can find in other lymphomas, but CXCR4 mutations, on the other hand, finding them like this was pretty unique to Waldenstrom. So this was, in fact, a major discovery, a seminal discovery, and it appeared as a plenary paper, as you can see there, in the journal Blood. Now, what this mutation does is quite interesting. There's this molecule called CXCL12, as you can see there. It's actually made by the bone marrow. And in turn, this molecule is free floating and it comes in contact with CXCR4, which is like a receptor, an antenna that sits on the Waldenstrom cells. So, in response to it binding to CXCL12, the cells begin to traffic and they start going towards the bone marrow. And that's why Waldenstrom's, unlike the other lymphomas that we talk about, is a bone marrow-centered disease. It's largely because of this type of trafficking. And about 40% of all patients have the CXCR4 mutation, but the rest of the patients that don't do something very interesting. They overexpress this receptor, so they're also responding to these signals by CXCL12 and migrating towards the bone marrow. Now, there are over 40 different types of CXCR4 mutations. Unlike in MYD88, where it's essentially one type of mutation and we can do a simple PCR test to show if somebody has it or not, here we have to use next-generation sequencing because, quite honestly, there's all these different mutations, and the most common of which is the one that I show you in that box called the S338X. Now, you might ask me, what does that mean? Well, what it means is that the amino acid position 338, there's a serine amino acid, if you remember all that from your basic biochemistry. S stands for serine. And what the X means is that at that location, the protein has been truncated or deleted. So that tail is now gone. And that's actually a very pivotal event for many of you, because what it does in turn is it distorts the signaling of this molecule, which in turn doesn't allow CXCR4 to shut off. And as a result, it stays on and continues to signal. And that signaling actually contributes to more IgM being made. For patients that come in with hyperviscosity syndrome, it's because of this signaling. And also it contributes to drug resistance, as our laboratory as well as others have shown. So it's a very important mutation to keep in mind. And this has also been factored into how people do on BTK inhibitors. And again, going back to that slide where we had trials that incorporated CXCR4 testing, what did we learn? Well, what we learned is that if you have the CXCR4 mutation, it's going to take longer to get to those deep responses. In fact, it's 4.5 months versus 1.9 months if you don't. Now, that's important to know. And you may ask me, well, why should I get tested for CXCR4? Probably that should be a reason of itself, because frequently you get doctors calling and saying, you know, I put somebody on a BTK inhibitor, they're not responding, or they're slowly responding, and I want to change, to another therapy. And that's the first question I ask is, have you checked the CXCR4 mutation status? Because it's just going to take longer, and you don't want to abandon what might be good therapy. The depth of response, on the other hand, isn't as decent as somebody who doesn't have the CXCR4 mutation. Look at that line that says median VGPR. It's 14%, which means those, over 90% reduction in disease burden was only achieved in 14% of patients versus 41% that didn't have the CXCR4 mutation. And what you also see collectively is that the duration of these responses, are lower for somebody with a CXCR4 mutation, 59% versus 75% for those that don't have it. And so it is really impactful. I do think it's important for us when a patient is diagnosed to get the MYD88 as well as CXCR4 mutation status. Now, the question that I sort of laid out there earlier for you was, why don't we see CRs? Now, what does CR mean? Complete remission. It doesn't mean cure. It just means that the way we go about evaluating response, checking for IgM, doing, bone marrow biopsies, doing CAT scans, if we're looking for residual disease, then it's still there. And that's why in the setting of even a complete remission, you're going to end up with this, somebody saying to you, you're in a complete remission, but if you really scratch very hard and use molecular testing, disease is still going to be there in the majority of patients. And so the big question is, why is it that we're not getting these complete remissions? And it has to do with these molecules called the IRAK molecules, IRAK. They're circled there in red. And there's two of them. There's Iraq-4 and there's Iraq-1. Now, these molecules are sort of a, backdoor channel to keeping the cells alive. You can see that they're independent of the HCK-BTK pathway. So we are drugging and poisoning that pathway, but on the other hand, the Iraq molecules persist. And this is important for us. This work was funded by the IWMF as well as LLS and the NIH, and I want to tell you this was also a very important revelation to us to try to advance therapy for Waldenstrom's. Perhaps there were people here in this audience who participated in the ibrutinib pivotal trial, which was the trial that led to the FDA approval. But what we saw, and it's outlined there in red, is even though we can shut down BTK, the Iraqs stayed on, and that's what those histograms are supposed to show you. And this is actually from bone marrow samples taken after patients were on ibrutinib for 6 months. So the cells were still alive, and we were trying to figure out why are they still alive. Well, it turned out that it was the Iraqs that were turned on. Now, we knew that the Iraqs were playing a role because we would take the cells then from patients 6 months after they were on ibrutinib alone, and then we would put them in the test tube and incubate them with both an Iraq inhibitor as well as ibrutinib, and we could kill them. So this was telling us that that Iraq signaling pathway was still on and maintained. So we began to work very hard trying to understand, how these Iraq molecules were actually keeping the cells alive. And this is, again, work that's been funded by the IWMF, which is why I want to show it to you. But what you see there in the first, on the left-hand side, is what happens to these cells if you knock out their Iraq-1 or Iraq-4 gene. And what you see is they're dying, right? All those lines are going downward. That's a good thing. Now, what's happening, on the other hand, as we discovered, and this was a key discovery in our lab, is that these cells begin to start throwing off these distress signals. Those are in the red circles that I show you there. So you see that the blotches there, that actually represents protein. And in this case, it's representing a form of the molecule ERK, which is a very important survival protein in Waldenstrom's. And you see that when you knock down either Iraq-1 or Iraq-4, these flares go up, and the cell is trying to stay alive at this point in time by using now this alternate pathway for survival. What's more interesting is that if you now knock down the Iraq-1 and you see the flare going up, this survival signaling, you can actually quench it very quickly by incubating those cells with ibrutinib or with an HCK inhibitor, which I'll talk about in a little bit. So you see all of a sudden in the circle there that ERK signal goes away. And this is what we're trying to accomplish now, is developing drugs that can actually extinguish out Iraq and use them in combination with the BTK inhibitors. And so what I want to do is tell you a little bit about the progress that we've made in overcoming this Iraq resistance to BTK inhibitors. And one of the smartest things that I think we did is we developed a dream team, or brought in a dream team. Some of you may remember the A-Team from way back. I used to watch it all together. These are my A-Teams, so you know. And they include some incredibly smart medicinal chemists. Nathaniel Gray, probably considered one of the top medicinal chemists in the world. He used to be at Dana-Farber and Harvard, and then he got homesick and went back to California along with his family. He's now at Stanford, still collaborating with us. And then Dr. Sarel Barlagi, who's at Harvard Medical School, Dr. John Hatcher, who's actually part of our center now at the Bing Center, and Dr. Gino Wang. So, what are they working on for us? Well, they're working on developing these drugs called PROTACs. And PROTACs stand for proteolysis targeting chimeras. It's always nice when we can abbreviate these long, acronyms. But these are very interesting drugs. And Nathaniel, as well as John Hatcher, have the unique expertise for making these drugs. And what these drugs are, very simply, they're drugs that bind to proteins in the cells, and then they drag those proteins to wastebaskets which exist within every cell. You have to remember that proteins are made every day, every moment, in our cells, And if they weren't broken down, they'd accumulate, and then what would happen is the cell would die because it would just choke on all this, all these proteins that are just accumulating. So there is a wastebasket that gets rid of proteins, but you can actually direct proteins in a very smart and targeted way to these wastebaskets by creating these PROTACs. And that's what we've been working on. In fact, we developed a first-in-class IRAK1, IRAK4 PROTAC. PROTACs are also known as degraders, as you can imagine, to degrade down a protein. And you can see there in the first, blot how this drug JHX 13-0501, we'll call it 0501 to keep it simple. It shows you how, once again, those protein blots are vanishing as we increase the drug dose, predominantly for IRAK1, but also IRAK4. And more importantly, in the histogram that you see next to it in red, that's when we can take MYD88 mutated lymphoma cells and combine 0501 with ibrutinib. And you see now the color red throughout those blotches. That means that every dose combination that we tried, the activity was synergistic. That's a good thing. So when you see red in these particular assays, it means you are killing those cells in a very synergistic way. And that's why we're excited about the development of this drug. We've advanced this drug class, again, thanks to the IWMF as well as LLS funding, to now make even more potent degraders, and ones that are actually starting to show even better activity when we put them in our animal models. Now, more excitingly, and this is actually new data, so you're only seeing it here for the first time, I haven't shown this anywhere else, is that when you combine 0501 with either zanubrutinib, pirtobrutinib, even the B1 drug, the BGB16673, I'm going to tell you about that, as well as the Nurix degraders, in all cases, what do you see here? The color red. We said red is actually really good. Now, why I'm really excited about this is it also represents an interesting new take for us, because we were always afraid that if we use two PROTACs, two degraders together, these might actually compete for that wastebasket. And as it turns out, even here using other degraders along with the 0501 drug, we're still seeing as much red as you can see here in these blots. That tells us that the wastebasket system is actually quite unlimited, and we shouldn't worry about combining these PROTACs together. And this hasn't been shown before, so this is why this particular blot is this particular slide is very, very exciting. This paper is now being submitted for publication, and IWMF as well as LLS are really the ones who have made the most difference in allowing us to move this field ahead. Now, for us, even a bigger concern these days is that patients are becoming resistant to BTK inhibitors. For some of them, it may occur within a year or two. For others, it may occur 10, 12 years later. So we always need to have action plans. And this is why we've been studying how resistance to BTK inhibitors is acquired. Now, when you use the word acquired, it means, whatever mutation was there wasn't there at the beginning. It just happens to show up if you have somebody on a particular drug. So the most common mechanism for acquiring resistance to drugs like ibrutinib involves having a mutation at the site where ibrutinib is supposed to bind to BTK. In the case of ibrutinib, zanubrutinib, acalabrutinib, even tirabrutinib, these are called covalent BTK inhibitors because they all bind to cysteine 481. So again, amino acid number 481, there's the amino acid cysteine there, and ibrutinib loves to go there and bind to it and shut down BTK. On the other hand, patients develop mutations at this site, and over time, these mutant cells become enriched. And I'm going to show you some data in just a second. And unfortunately, if you develop a mutation there where you switch the amino acid, in this case, serine is the most common amino acid switch, so you go from cysteine to serine, all of a sudden what you see is that the ibrutinib can't bind at that spot anymore. Now, Leanne Zhu, who was with us for quite a long time, and she's really the mother of MYD88 testing. So I want to tell you, once MYD88 mutation was discovered, she was the one who went to work and developed the first PCR assay. So we owe her big. And in fact, her template is used almost worldwide for the detection of MYD88. There are newer platforms now, but hers was really the one that's used in most centers. She made an interesting and startling discovery, and that is that in patients with Waldenstrom's who developed resistance to ibrutinib, it wasn't just one mutation that took over, and populated the cells, but in fact, in some of these patients, she found 4 different mutations. And this is important because what it means is that from the get-go, at a very highly microscopic level, these BTK mutated cells exist, but there are just so few of them that you can't pick them up, even with very good molecular testing. But over time, what happens is that you begin to enrich these. These become the predominant cells. And she showed in this particular patient that, at baseline, which meant at the beginning of therapy, you couldn't pick up any of these mutations. By month 11, again, nothing. It's 0, 0, 0 for the 3 types of mutations that she was following. But by month 22, you can see that 2 of them are starting to show up, and they're still at very, very low frequencies. But look a year later, in fact, all of a sudden, these are just springing out like weeds. And this was actually very telltale because it shows you, as we see with other cancer evolution, that once you find a cell that can avoid a particular therapy by mutating its contact point, then all of a sudden, over time, like weeds, it begins to grow. There was also another part to this that was actually quite fascinating for us, and that is this mother and son team. Dr. Chen is the son that you can see here. Dr. Leanne Zhu underneath is his mom. The 2 of them worked together to be able to profile exactly what these mutations were doing. And what they found in this particular case is that if you acquired the mutation, the BTK CYS481 mutation, and that's what the little red arrows are showing you, all of a sudden that ERK pathway that we just talked about also begins to light up. So once again, these cells are now trying to use the ERK pathway in order to survive. Now, this ERK pathway, you may ask me, what is it doing exactly? What it actually is doing is it's firing up the cells to make all kinds of inflammatory proteins. And these inflammatory proteins are what mediate a lot of the survival signaling. They actually turn on the microenvironment that the cells are living in to help protect them. And just look at the difference between the 2 cell lines here that we had. One is without the mutation, that's the WT wild type. The other is with the mutation. And you might notice that there are some color differences for all these inflammatory cytokines. And that is that the orange and yellows are starting to light up in the ones that are mutated. That just means that inflammatory cytokines are being secreted. But then came the most interesting and startling discovery of all, and that is these bad cells, these BTK mutated cells, were actually talking to their counterpart cells that didn't have the mutation, and through these inflammatory cytokines, particularly interleukin-6 and interleukin-10, they were causing these cells to become resistant to ibrutinib. And we prove this by having these co-culture experiments where you use a membrane to separate 2 different cells. You can see in the first instance that these are cells that are wild type. You see WT on both sides of the membrane. You add ibrutinib, they all die. Well, that's a good thing. But then the second set of experiments that you see, you have the cells that are mutated on top, and you have the wild type cells on the bottom, and you're adding ibrutinib, and guess what? They're surviving on both sides of the membrane. So even the cells that don't have the BTK mutation are surviving. And so it's because of the interleukin-6 and interleukin-10, these are again inflammatory proteins being released by the mutated cells. And in the case of IL-6 and IL-10, you can actually use antibodies to neutralize them. And then what happens is the cells that are wild type die, but those that have the mutation continue to survive with ibrutinib. So there is this crosstalk that exists that these bad cells are corrupting even the good cells because of these inflammatory proteins that are being released. So how can we reverse this? Well, there is second now and third generation BTK inhibitors that shuts down this signaling pathway. And Dr. Maneet Munshi, before I can call him now doctor because he's done with med school, but he was in our laboratory and did these very elegant experiments where he treated cells with pirtobrutinib, which is a BTK inhibitor that binds to a different site on BTK itself. So even if you have the cysteine 481 mutation, this BTK inhibitor binds in an alternative location, and guess what it does? It shuts down that ERK signaling pathway. And this was also true in mice that were, engrafted with these tumors. And so this actually gave way to the study of pirtobrutinib in previously treated Waldenstrom patients. This was actually led by my friend and colleague Leah Palumba at Memorial Sloan. And our paper just got published in Lancet Hematology. Many of us in this room participated in this study. But even among patients that have already seen drugs like ibrutinib and zanubrutinib and calibrutinib that progressed, pirtobrutinib, shows very high levels of activity and even sustained activity. So this is a good thing. It means we now have a plan B to go, for those patients that are progressing on the covalent BTK inhibitors. Now, there's also a new drug that I want to share this exciting information with, and this is the drug BGB-16673. I alluded to this a little bit earlier. This is one of those PROTACs or degraders. And what this drug does is it binds to yet a different site of BTK, and it's able to this time not just shut down BTK, but actually degrade it altogether, destroy it. And so my colleague and friend, Dr. John Seymour from Australia, has been leading this trial. And again, very high response rates. We're seeing, almost 80% of patients who have received this drug, showing a major response. And despite, whether they're BTK mutated, MYD88 mutated, CXCR4 mutated, or even TP53, which is a negative prognostic marker, in all these cases, the response rates are quite high. So this drug is currently in development. I do expect that this will be approved at some point in the near future. But again, it provides us now even a plan C in the BTK world, which is very, very important to consider. Now, this slide is meant to bring us back to reality, and that is that no matter which of these drug classes we're looking at, the covalent is represented by the ibrutinib, zanubrutinibs, non-covalence, which is the pirtobrutinib class, and PROTACs, which is BGB-16673. You see, in fact, mutations in BTK that can lead to resistance. And that's why we need now a plan D, because these mutations will invariably, after perhaps a lot of time going by, will start, becoming problematic for us. And that's why, thanks to the IWMF and LLS and the NIH, when the NIH was still with it, these days it's a different story, as you know, but we had funding to actually develop targeted therapy against HCK. Remember in that cascade that I showed you, at the beginning, MYD88, upregulates and turns on HCK, and it's HCK that actually turns on BTK. So what we've been trying to do is to go after the enabler of BTK itself. And we've made remarkable progress here that I'm really excited to share with you. And that involves the development of a particular HCK BTK degrader. And this was also accomplished with the help of the medicinal chemistry team. And this molecule is known as DFCI 0206. I'll call it 0206 to keep it simple here. I'm very excited about this molecule because this actually does a couple of things. It still retains its ability because of the initial scaffold that we built to shut down the activity of HCK as well as BTK. So in this regard, it acts like a kinase inhibitor that just shuts down functionally the operation of the molecule. But more importantly, it also degrades it. So we get really a twofer here: kinase inhibition and degradation. And what you see in the first graph there is that when we administer this drug at different doses, we actually stop tumors from developing in the mice that we engraft these MYD88 mutated tumors. They stay stagnant and don't grow. And then what you see there is the tumors being taken out 6 hours or 24 hours at the end of treatment. And you see, in fact, that we're able to fully degrade BTK in the tumor by giving the drug by mouth in these mice, and HCK as well is degraded, and this process can last well over 24 hours after we've given the final dose. Now, I'm excited about this molecule because when we compare the activity of 0206 against drugs like zanubrutinib and even the scaffold that we built this on, KIN 8194, again, that was thanks to IWMF support. This drug actually shows higher levels, significantly higher levels of activity in primary Waldenstrom patients' cells. And so this experiment there involved 13 different patients and looking at how this drug acts in those compared to zanubrutinib and others. So I'd like to tell you this now is a drug that we're moving into IND enabling phase, and I hope in not too short time we will have this we'll have this drug in human trials. So it does really represent the ability to go from bench to bedside. Now, the next set of drugs I want to talk about, because these also represent drugs that we use in people who progress on the covalent BTK inhibitors, the non-covalent, as well as the degrader BTKs, involves BCL2 inhibition. Now, if you know nothing about BCL2, that's totally cool. BCL2 is very active in most of our cells. It protects cells from dying, and in some cases, that's a good thing, right? But in the case of Waldenstrom's, it's a bad thing because we do want those cells to die. And even if you throw chemotherapy or BTK inhibitors at these cells, this BCL2 molecule acts as sort of a deflector shield. It protects the cell against these. You might think about this in the context of like a good soccer match. You have a goalie that's constantly blocking, that shot from entering into the net. And that's how I see BCL2. But luckily, we actually have a couple of drugs that are very active and can shut down BCL2. One you may have already heard about, venetoclax. This is a drug that many of us have worked on, and now there's a new drug, sonrotoclax, which is also being developed by B1, and I want to talk to you about some of the results with these drugs because it is quite exciting. So my colleague and friend, Jorge Castillo, led the venetoclax trial in previously treated Waldenstrom's patients. And this was a very exciting trial. We published it in the journal JCO, which is the top oncology journal, back in 2021. Some of you may have participated on this trial. We were only given drug to give to patients for 2 years. Many other studies gave the drug indefinitely, and I think this could have perhaps impacted some of the outcomes. But nonetheless, over 80% of the patients had a major response, and it wasn't impacted by the CXCR4 mutation. And so this actually represents a very important, drug. We did see neutropenia, which means the lowering of the white blood count in quite a few patients. So the original dose that we were using was up to 800 milligrams a day and giving the drug for up to 2 years. But most patients actually tolerate 400 milligrams a day, and that tends to be the dose that's being used these days. But nonetheless, showing us that you can actually get long-term disease suppression. In this case, the progression-free survival is 30 months for patients that actually got 2 years of therapy. Now, in Spain, my other good friend and colleague, Dr. Garcia Sanz, has been leading the efforts with sonrotoclax. This also has a BGB name, BGB 11417. This is still an evolving trial, but nonetheless, up to 80% of patients had, at least an overall response, and many of these responses continue to be durable, meaning they're still lasting. This drug is a little bit easier to give than venetoclax. You can actually ramp up because in all these drugs you go a little slow. You give a little bit of drug, then you go up, and then you wait, and then you go up a little more, because when you hit BCL2 hard, the cells can die very fast, and that creates a mess for the body because it starts reacting to all these dying cells. And that's why giving sonrotoclax may have an advantage in the sense that you can actually ramp up the dose in a faster way, which is good for patient care. Now, back home, we wanted to see what would happen if we would take a drug like pirtobrutinib and combine it with venetoclax in patients. And years ago, in fact, back in 2015, we were the first to publish ever for any indication that if you added a BCL2 inhibitor like venetoclax along with a BTK inhibitor, you were going to see synergy. And that's why we were so excited to do this trial. I'm sure, again, there's people in this audience that are on this clinical trial, but we were very eager to test this in previously treated individuals. And so this is only a snapshot of the data we presented at the American Society of Hematology back in December. I'm very happy to tell you this trial is now fully accrued, and we're actually hopefully going to have an update of this at this year's American Society of Hematology meeting, as well as the International Workshop on Waldenstrom's. And what's exciting there is everyone responded. Major responses occurred in 96% of patients. And I think that VGPR rate that you see there is going to creep up. In fact, we've also had people who have attained a complete remission. And again, it's early to take snapshots of how this is going to behave. But at 1 year, 94% of the patients still remained, in a remission that had, attained a response. And so I'm excited about this. I think it, has the potential to be a new standard of care. But we do need, more follow-up to see what the relative benefit of combining these drugs is versus the single, the drugs being used singly in relapsed or refractory Waldenstrom patients. Now, we were in Athens last week. If some of us look a little blurry-eyed, is because we've been going back and forth over the Atlantic. But there's a lot of excitement, even on the other side of the Atlantic, that I'm going to share with you. This was the European Consortium for Waldenstrom's Dr. Christian Buske, who's here with us. Is he in the audience here? There he is, yes. A round of applause for Dr. Buske. A round of applause because not only for the incredible work that he does, but again, we were together in Boston at the end of March. We were together in Athens just last week, and here he is now, quite a trooper, back in Columbus, Ohio for the IWMF meeting. But the ECWM is a tour de force. As you can see here at the Acropolis, that we all had a chance to have a group photo. And it's quite a tour force because it's doing really the big trials. You do see innovation in all the drug discovery on both sides of the Atlantic, but the big trials that are actually helping cement their use as standard of care, a lot of that, and I would even say the majority of that, is being done by the ECWM. Now, why I show you that picture first is because in Greece, in the old days, they had this concept of the clepsydra. And in fact, have any of you been to Athens? Yeah, a few hands. There's the Agora, which represented the old gathering point or marketplace in Athens. But there was where also people gave speeches. And you were allowed to speak as long as you were a citizen, but you were timed. And the way you were timed back then was through the clepsydra. It was basically a water clock. As soon as all the water ran out from the top jug into the bottom, you were out of time, which is probably where I will be in a few minutes' time here too. But, when I think about all the current therapies that we use for Waldenstrom's, I think of it in the context of clepsydra, because what we're doing is we're giving these drugs, and then it's just a matter of time before, the benefit runs out. And then we go on to the next therapy and the next therapy and the next therapy. That's good. And I'm not going to say that's bad. It's good. But perhaps a new way for us to challenge ourselves is to start looking at other ways to treat Waldenstrom's, even with a curative intent. And this is why I was very excited about the conference on cellular therapies that we had for Waldenstrom's in Boston on March 28th and 29th. Many of you know these therapies are completely changing and revolutionizing our therapy of lymphomas as well as multiple myeloma, where they're not talking even about CRs, they're talking about MRD. MRD means we're using molecular techniques and we can't find disease. We can't even find 1 in a million cells that actually have disease in the bone marrow of myeloma patients because of cellular therapies. And you hear at all the different meetings that you go to that, I had a Waldenstrom patient who had, high IgM, had lots of disease. I gave, a CAR T cell therapy, which is a cellular therapy, and 28 days later, there was no evidence of disease. And so when you start hearing stories like that, you realize this therapeutic approach, really harnessing the immune system, could be very good for us and in fact may get us to those cures that we want. And that's why this was such an important meeting. You can see we had 42 speakers, and the IWMF was well represented. Chairman Kitchen was there, President Delora Senf was there, and we also had David Schenkein, who was also there, who helped us with the advancement. And so what we learned, and I also have to credit Tim and Jenny Bliss for funding this, and not only funding this meeting, which brought together academics on both sides of the Atlantic, but it also brought drug companies together. If you can just imagine this, because usually these kind of advisory boards are funded by industry, and you never really have industry talking to industry. In this case, industry was helping each other in terms of how do we develop these drugs in the setting of Waldenstrom's. And that's what Tim and Jenny Bliss accomplished for us. But on top of that, as you will probably hear at this meeting, they also made a very large commitment for a gift in the millions of dollars to help advance these therapies for Waldenstrom's. And the IWMF will be supporting and administering the RFPs for that particular fund. Now, what's included in these cellular therapies is kind of an umbrella term, are the bispecific antibodies. These are antibodies that do something very interesting. They grab onto the tumor cell, but they also grab onto the immune cells, in this case T cells, and they bring those together. And because of that interaction, the T cells end up destroying the tumor cells. In this case, I'm showing you an example of teclistamab, which is an antibody that binds to BCMA, a target on multiple myeloma cells, but on the T cell it binds to CD3, and because of that interaction, the T cell is able to kill the tumor cell. Now, you've also been hearing in the news about CAR T cells. There's many different varieties of CAR T cells. I'll allude to some of them in just a second. But this is a more interesting and a little bit more complex process where you actually take the T cells and you end up transfecting them in such a way that you're now putting the target that you want to go after on the malignant cells, and then you reinfuse those cells back into the patient, and those cells go find the tumor cells and destroy them. And this is a very potent, approach to killing cells. Now, the Waldenstrom clone, on the other hand, is a little bit complex, because when you think about Waldenstrom's, it's not just one cell type, which is the case in myeloma or most lymphomas. It's actually made up of 3 different cell types. It involves B cells, much like lymphoma cells. It involves these lymphoplasmacytic cells that are sort of changing or morphing into plasma cells. And plasma cells are what you see in multiple myeloma. And the relative proportion in Waldenstrom's is 5 to 10%, 5 to 10%. So most of the cells, most of the reservoir is in the B cells, but the plasma cells at the very end make up about 10 to 15% of the clone. And those are the cells that make the IgM. So you really do want to go after the entire clone if possible. So I want to credit here about both Dr. Howe Sun in our group, as well as Zachary Hunter, who worked together to help us at least create the first map of targets that we can go after with these cellular therapies. And just to exemplify a few, I'm only going to exemplify the therapies that we discussed in Boston. But the first one is CD19. CD19 actually is on the B cell compartment. And as you can see the little graph underneath in pink there, those bar graphs, you'll see in fact that the malignant B cells of Waldenstrom express it more highly than plasma cells, but they both do in fact. And there is an exciting bispecific antibody. I think Dr. Tarek Muhyiddin is going to be talking about this. It's made by AstraZeneca that targets CD19. But we also have all these CAR Ts that were represented at the meeting, including one that targets CD19 and BCMA. There's that's from AstraZeneca. There's one from Kite Pharmaceuticals that targets CD19 and CD20, which I'll show you in just a second. And there's also what they call allo-CAR Ts. These are somebody else's T cells that have already been made into targeted therapies, and they're then infused into the patient. CD20, probably most of you know about because rituximab goes after CD20. And there's actually a couple of very interesting bispecific antibodies. Epcoritamab, there's a clinical trial now, and I think you're going to be hearing about this with the WMNet reporting. And there's also mosunetuzumab. The epcoritamab bispecific trial is happening on both sides of the Atlantic. And the CAR T that I mentioned earlier, Kite 753, made by Kite Pharmaceuticals, targets CD19 as well as CD20. And again, look at the bar graph with the pink bars. You'll see, this target, CD20, is mostly found on the B cells. And so this is why we need to keep in mind the entire clone, as I mentioned to you. So BCMA, on the other hand, is the target that's revolutionizing myeloma therapy because it's it's very highly expressed. There's a number of bispecifics. You're going to be hearing about them. In fact, Prashant Kapoor is going to be talking about his trial with elranatamab. And Andy Brannigan, shown there, is going to be talking about his, or WMNet talk will be about teclistamab as well. And then there's all these CAR T cells that have also been formulated. The one that we're most excited about is the AZD120 because it hits 19 as well as BCMA. So this is actually an example of a CAR T cell therapy that can overlap the entire Waldenstrom clone. And so I think, and there is now a lot of interest by AstraZeneca to help develop this drug for Waldenstrom's macroglobulinemia. And again, thanks to the prowess of Zach Hunter, we even have now a new target, and that involves FcRH5. This was analyzed from our initial genomic sequencing data. There is a bispecific antibody called cevostamab that targets this, but what I want to tell you is it's strongly expressed on both the malignant B cells as well as the malignant plasma cells of Waldenstrom. So here we actually have a twofer in one particular target. And Zach's prowess here has shown us that it's very different, that FcRH5 is very strongly expressed in Waldenstrom cells in deference to normal B cells. So this is really a standout target. And again, it shows you how the genomics are going to help us advance to new therapies. Now, one other thing that I just want to say as I'm closing out here is that all that we have learned with BTK inhibitors will come back and potentially benefit us with these new cellular therapies, because Dr. Howe's son, and others have shown us that you can actually excite the immune system and make all the bad, exhaustion of our immune system go away by using drugs like ibrutinib. And he showed this very nicely in a paper that was just published in the journal Blood, the highest impact factor hematology journal. So I think he also has presented to you his work. But I want to say this is perhaps one of the strong studies telling us why BTK inhibitors with these cellular therapies have a future. You also, heard from, the Italian twins here, Dr. Montello as well as Dr. Guerrera. Their work actually is focused on these cells, which we call the myeloid-derived suppressor cells. These are cells that are being duped by the Waldenstrom cells to actually secrete proteins that then shut down the immune system. And there's these molecules called the S100s that appear to be doing this, as Dr. Guerrera from our group has shown, to mediate this exhaustive profile. So again, this is going to be very nice work, that both of them are working on right now to help reverse this immune suppression. So I just want to say one of the reasons why I loved going to Athens was to hear about the exciting trials that are being planned by the ECWM. And when we had actually our panelists from both the US and Europe meet, there was so much excitement about the use of the BTK inhibitors along with bispecific antibodies, and a trial is currently being planned by Christian and his group. So I'm very excited to see that. And hopefully, as is planned, it'll be a randomized trial, so we'll see the relative benefit of using a BTK inhibitor with a bispecific. So I just want to conclude here by saying there's lots of progress. I tried in an hour's time. I did make my time, so the clepsydra should be happy. It's not dried out yet. But there's a lot of progress to report. And what used to be, for us a one-day meeting now is 3 full days. And even then, I don't think we'll be done with reporting all the progress from all these very exciting innovations and clinical trials. I would be very remiss if I didn't thank all my collaborators back on the home front, as well as the various Harvard laboratories and external collaborators. Jorge Castillo leads our clinical group. And as you can see, Shana Sorosiak, Tarek Muhadeen, and Camille Edwards at our center, Andrew Brannigan at MGH, and Gottfried von Kudel help us with advancing these studies, and many of the other laboratories that help help us advance these new therapeutics. And I just want to conclude by thanking all our supporters and funding agencies. You can see there's quite a few, philanthropy foundations that help make this work possible. We're not counting very much on the NIH these days, but the IWMF and LLS and WMFC have been there for us. And I'm going to just leave you with this last slide here because I think it really summarizes the spirit that we had in Athens. And that is that what you leave behind is not what is engraved in stone monuments, but what is woven into the lives of others. And this is one of the famous quotes by Pericles. And in that regard, I want to thank the IWMF because what you're doing here, along with all our patients, is being woven into the lives of others. And with that, I want to thank you for your attention. Thank you.
Dr. Treon has agreed to stay for a few questions if we have some, and he's never not had questions, so this would be a real first.
Here.
Thanks so much for all of that. It's quite overwhelming. I'm just curious as to whether or not there's any way to predict the effectiveness of any of these new therapies for extramedullary Waldenstroms.
Yeah, it's a great question, Eileen. And I think this is where cellular therapies will play a particular role. Now, the BTK inhibitors do also show, benefit on extramedullary disease. So do the BCL2 inhibitors. You do see reduction, even elimination of tumor cells to the point that you can't pick up, by CAT scan, extramedullary disease. But I think cellular therapies in particular are going to be very exciting in that regard because, we're seeing patients who have PET CTs that are just chock full of tumor and with lymphomas. And, weeks to a few months later, all of that is gone. And even the PET scans are showing up negative. So this is why I think advancing cellular therapies is indeed a priority, particularly for extramedullary disease. Now, when you say extramedullary disease, that term really means outside of the bone marrow. And of course, the other area that we're always concerned about is Bing-Neal syndrome, when you have disease in the central nervous system. In this regard, the BTK inhibitors have truly revolutionized, therapy. They're able to get into the, through the blood-brain barrier into the central nervous system. And it remains to be seen what the cellular therapies, can also contribute. There have been, reports as well of benefit there. But I still think, BTK inhibitors and even new generation BTK inhibitors will still be very important for us to continue to develop for Bing-Neal syndrome.
I have one here.
Thank you. This is a real primitive question. When I was looking at your graph and I was looking at everything that was downstream from the MYD88 mutation and looking at Iraq and looking how you're attacking the BTK mutations, my first type brain says, who's going after the MYD88?
We are. Yeah, we have made considerable progress in being able to degrade MYD88. I'm so glad that you asked it. I think it's probably a little premature to get overwhelmingly excited about this, but one of the grants that we have from the IWMF has been to develop directly a MYD88 degrader. And so we've synthesized the protein, both the mutant protein and the wild type protein. Wild type just means the normal protein. And we ended up using the prowess of one of our proteomics groups that found all these binders against the MYD88 molecule. And then from there, we were able to build degraders. And one of our degraders, DFCI something 23, O223, is showing incredible activity, particularly against the mutant form of MYD88. It's able to degrade it. It's actually able to destroy the Waldenstrom cells from patients as well as the cell lines. But we're still profiling it. We want to see whether or not it's absorbed and develop some data around the pharmacokinetics of the drug. But stay tuned. Perhaps if Chairman Kitchen invites me back next year, I'll have more to say about that.
I think you can count on getting it in.
Yeah, I hope so. I can't presume.
That's pretty safe. We're also going to get to see Dr. Treon tomorrow morning and have his wisdom. Whoops. There's another question.
Is there any value in treating someone who is asymptomatic, who has extramedullary disease, by treating both the MD88 the bone marrow and the extramedullary?
Yeah, that's a complex question because you really need to know the patient and see what's been happening to the patient. So if somebody's asymptomatic, in general, we try to hold off on therapy until, that patient meets sort of the expected guidelines that we have for treating individuals. And that really is a tradition that goes back to Jan Waldenstrom, who used to say, let well do, don't interfere. Now, mind you, in Jan Waldenstrom's time, therapy was quite limited. And even in my time, when I first started out, we had chemotherapy, and in some cases, we had really some horrible side effects with the chemotherapy we're using. But now with more modern therapy, that sort of when to start, when to hold is changing. The mentality in that is changing. In either case, we don't want patients to fall off the cliff before you extend a helping hand, but you really need to know the patient and know, at what rate they're progressing, if they're progressing, because that can be quite variable, and to see how symptomatic they are. Extramedullary disease, on the other hand, we do pay attention to. So if we do see that lymph nodes are growing and the spleen's growing, we tend to be a little bit more responsive to getting therapy going sooner than later. But I don't want to answer in your particular instance because you really do need to synthesize a lot of information before you make that recommendation.
Yes, this is the last question.
All right, we have a question right here.
So this may be a little bit related to the previous question, but do you know how to prevent the gene transformation, like for instance, from the cysteine to serine? Do you know how to prevent that from happening?
Yeah, it's not preventable because for many of the patients, these cells already pre-exist. Now, how can we alter the course? We just had a paper out in the journal Blood Advances, and it was a beautiful project that I gave to one of our graduate students, Dominic Pizzarella. And Dominic actually started with us as a co-op student. And, you know, he did beautiful work in our laboratory. He was, from the get-go, a star. He had to decide between MIT and Stanford where to go for his PhD, which was a very difficult decision. Anybody want to guess where he went? Oh, no, Stanford. I heard MIT. But he's at Stanford now with Dr. Nathaniel Gray. Now, he did a very interesting study because he theorized that if I use two BTK inhibitors together, in this case, he used zanubrutinib along with pirtobrutinib, that by binding to two different sites on the BTK, he could shut down BTK and not really risk that one particular mutation event is going to allow that clone to grow. So that combination we call now the ZAP combination, zanubrutinib, pirtobrutinib. I'm excited about that because I think it has the ability to suppress the acquisition or the enhancement of these mutant clones that probably pre-exist. Interestingly, when we looked at ibrutinib with pirtobrutinib, it was antagonistic. When we did acalabrutinib along with pirtobrutinib, it also was antagonistic. So there's something unique about zanubrutinib being able to interact with pirtobrutinib that allows for this benefit of ZAP. I'm hoping that this will translate into a clinical trial because we would all love to see ways that we can actually suppress the development of these clones. But thank you for asking that because that paper I just have the proofs in hand upstairs in my and my hotel room, just went through them. But that's going to really make a lot of us think about using these drugs together.
Thank you very much, Dr. Treon. It is a great privilege to listen to you and all your knowledge and your experience. It has been thrilled for me to be in Boston with Dr. Treon and being in Greece and just see all the researchers around the world who are working for us. I don't think the sun ever sets on people who are researching WM, and it's because of the IWMF and because of your contributions and support. And I know that Dr. Treon's talked to me about functional cure, and we're going to get there, and we're a partner, we're under the same tent, and we all want the same thing. So thank you for your excellence.